And I want to thank everybody. I know that we have a small audience right now. I anticipate that more will be joining. I want to ask that if you are not one of our panel, that you actually turn your video off. And that way the panel will show up on everyone's screen as being up on top. And I thank you for that. And we have with us today, beside myself, Dr. Garima Gupta, who is one of our faculty and is an intensivist. We have Dr. Evan Hermel, who is one of our faculty and lectures on immunology and all things germified. And we have Dr. Priya Nand, who is part of the Touro University Medical Group, is an infectious disease specialist and is practicing in Stockton. So I thank you all for volunteering your time for this. This is going to be more like a question answer format rather than a discussion with lecture. And what we've all done, I collected questions and I passed them to our three panelists yesterday in order to prepare for today. However, if while we're discussing things, there are more questions that come up, please don't hesitate to hit them in the chat and I will be monitoring those as well. And also, I've let our panelists know that most of the audience will probably be lay public as opposed to members of COM, for example, so that the lecture is directed more, or their answers are directed more to the lay public than other physicians. So what I want to start with is I had labeled some of the questions that I have from everyone. I got a dozen questions. I tried to group them together, and I made my own labels to ask people. However, after the person that I direct to speaks, if anybody else on the panel wants to add anything before I go to the next question, I'll leave it, I'll open up to the other panelists. So starting with Dr. Hermel, the first question, and I apologize, I have a second screen, so that's why I'm cheating off to the side, is there's some concern over the vaccine because it took less than a year to develop. And can you address the rapid nature of the development of this vaccine since it was less than a year from infection to delivery?
Yeah, so the speed in which this vaccine came into being was because the front end was greatly shortened by the fact that it's an mRNA vaccine. When you have a vaccine, you have to consider what goes into the development of the vaccine. You have to be able to first isolate the pathogen. You have to be able to then grow that pathogen in culture for one of two reasons. You want to grow it up in large enough amounts that you could kill it and then give a killed virus vaccine, or you have to attenuate it. This is an even longer process. You have to be able to adapt it into culture so that it loses its virulence, but still remains immunogenic, and then get it to a point where you could get it to a large enough amount to develop a vaccine. That's all eliminated by the fact that it's an mRNA vaccine. It took the Chinese, after the SARS virus broke out in Wuhan last year, it took about a month for the Chinese labs to sequence the entire genome. It took then about another month or two at most for the pharmaceutical companies to jump in and develop the mRNA vaccine. And what they homed in on was the spike protein, because that was realized very quickly that that was the major virulence factor and the determinant of the virus that provoked the best immune response. So you eliminate some 4 years of development by getting rid of all of the viral culture. Now, you have to also remember that the first immunizations went out in April, May. The emergency use authorization came out in December. That's not a long time, but that doesn't mean that it's not safe, because it went through the requisite phase 1, phase 2, and phase 3 trials. The collective amount, and I have this on a let me call up my slide here, if you will. Share screen. Okay, I trust you can all see the screen now.
Yes.
Okay, so this is an explanation of what the vaccines are. And for my students, you remember Dr. Gockin's lectures. It's an RNA vaccine. It can't do anything to your DNA. RNA stays in the cytosol where it gets turned into protein. Concerning safety, all right? Look at the numbers of the total people, the aggregate of people who were involved in the clinical trials, over 100,000. That is a good safety profile. In going to, I believe, a question raised by Dr. Russell, what's in the vaccine? You know, messenger RNA and several lipids which mimic the cell wall, which it's a lipid coat. It's essentially a glorified soap bubble, which allows the mRNA to get into the cell. It's a delivery vehicle. The data suggests that the anaphylactic reactions or the allergic reactions are being developed against the polyethylene glycol component. Anything else? We also know now that the allergies that are popping up are about 3 times. Let's see if I have the papers here. Yeah. So we know that anaphylaxis is a known side effect of vaccination. The rate of anaphylaxis and severe allergies are popping up about 3 times that of normal with this vaccine. So that's why everybody has to now sit for 15 minutes after receiving the vaccine to make sure they don't go into anaphylactic shock, and then you bring out the EpiPen in case they do. So where they rush to, on this slide is basically what I just told you. I also want to point out that the first mRNA vaccines were used in 2008, actually from a clinical trial for cancer vaccines from the year about 2005. First publication was 2008, and this was in a prostate cancer vaccine. For infectious disease, the first mRNA vaccine as a disease preventative was a rabies vaccine done in reported in 2017. So there was like a good 3-year period, I think, from the very first note that phase 1 trials had started, at least 3 years before that. The Pfizer vaccine, the Johnson & Johnson vaccine is a modified adenovirus, which is infectious, and it can get into a cell, but it is not replication competent. It cannot make more virus. All it could do is get into the cell. The virus is essentially a delivery vehicle, just like the lipid coat for the mRNA vaccine is. So the modified adenovirus vector, which has had its virulence genes knocked out as well, delivers the mRNA for the spike protein into the cell and makes the spike protein. We've been using recombinant viral vaccines in humans at least since 2006 with the Rototec vaccine, which is a recombinant bovine rotavirus vaccine, which is modified to express 5 human rotavirus proteins to prevent infantile diarrhea, severe infantile diarrhea. Okay, I think that's about it. I think that should answer the questions posed to me, at least the first 2.
So the virus, let's try that again. So the vaccine is getting into the cell of into what cell? So just very simplistically, if you can say that. And then it is producing antibodies to the spike protein of the virus so that if I get exposed to the virus, my body is able to fight it. Okay, so what cell is the shot getting into? And other than just making copies of attack the spike protein, which is basically what these vaccines are doing, is going on inside these cells. And I'll open that up for anybody.
Think about where the vaccine needle is going. So it's going into muscle tissue. Within these tissues, there will be resident antigen-presenting cells. They float around and they hit upon anything that is foreign. The vaccines have been modified. The mRNA vaccines are actually modified in a way that they do not provoke an innate immune response to viral messenger RNA, which would kind of like prevent the adaptive immune response to produce antibodies to the spike protein. It also allows them to be more stable. But so you have at minimum 2 different cell types that are expressing the spike protein. One would be muscle cells, and then antigen-presenting cells, dendritic cells, which are very well adapted to present the spike protein to T helper cells, which then will allow B lymphocytes to make antibodies to the spike protein.
Okay, thank you.
I think the only other thing I would add is in order for it to get into the human genome, the mRNA, that gets in through the vaccine has to actually get into the nucleus of the cell, and the mRNA does not get into the nucleus. So the concern about it getting into the human genome is not valid because it doesn't even get to the location where the genome is made, which is the nucleus of the cell.
To follow that up, you would have to even add another step beyond that.
Okay.
The messenger RNA not only would have to get into the nucleus, it would have to be converted at some point. It would have to be converted by reverse transcriptase into a cDNA, and then get into the nucleus, and then insert it into the genome. This all takes a whole series of steps and proteins, which were evolved by retroviruses to accomplish this. So this literally is impossible for just an injectable messenger RNA. The idea that the vaccine is altering your DNA is anti-vax propaganda, pure and simple.
So I'm going to keep us away. Excuse me just a second, Greg. I'm going to keep us away from any political speak. But what's important to know then is, for those of you that are not science-based, is that you've got this messenger that says do something. And for it to affect a human cell, it has to be read, transferred into the nucleus where it is read again. It then there's a whole bunch of steps of zippering and unzippering messages that can't happen because this vaccine does not get into the nucleus of the cell where the messages about what the cell is supposed to be doing occur. And if I am not languaging correctly, then I would hope our panelists will correct me. I'm going to keep going. Dr. Geyer, I know that you had a comment, but if you can write in a chat, because we have 9. Go ahead. Go ahead and just type it in there, Ariel.
I'm sorry, I'm so sorry to interrupt. I do want to add to the original question was about the speed of the vaccine manufacturing. There are a couple other, certainly the mRNA technology, which I think Dr. Hermel sort of covered really well, is one factor as to why it was easy is perhaps not the right word, but it wasn't, the challenging part wasn't making the vaccine. The challenging part was studying the vaccine. And there were some important differences in how this vaccine was studied compared to traditional vaccine science, which I think come up a lot in the news. So I did kind of want to address those as well in terms of speed. So one thing that I think we all have to keep in mind is, you know, we always say follow the money, right? So much money from the public and private sector was pumped into this operation. So all of these companies started working on these vaccines pretty much simultaneously. As the genome was published, they were able to get a vaccine candidate created and ready to study within a matter of, you know, weeks, I'd say a month. The other thing is that the government, the United States government subsidized subsidize the research, development, and manufacturing of the vaccine. So normally what you would do is you do a phase 1, you'd wait 2 years for safety data. You'd do a phase 2, you'd wait 2 years for safety data. You'd do a phase 3, wait 2 years for safety data. We're already up to 6 years. Because of the amount of money that was pumped into this, they could essentially do these trials back to back to back, one flowing into another. And the government subsidized the manufacturing. So before we even knew if the vaccine worked or didn't, the companies, Moderna, Pfizer, Johnson & Johnson, all of these that you're hearing about, AstraZeneca, they were all mass producing this vaccine and storing it. So as soon as we had an answer that it worked, as soon as the emergency use authorization was issued, the vaccine shipped the next day. Normally, you would finish all of the, you know, all of your research, you'd publish it, you'd get peer review, and then they'd say, okay, let's start manufacturing it now. So that entire timeline was shortened primarily because of the immense amount of money that was poured into this and the support that these companies had from the public and private sector. They did not have to take on any of the risk. If it didn't work, we just wouldn't use it. But that was part of that pandemic response. The other thing they were able to do is shorten that 2-year follow-up for side effects. So normally you would wait 2 years for safety data. They waited about 2 months. Now that sounds scary because you think, well, what about the rest of those 2 years? What are we going to find? If you look historically at vaccine side effect data, most side effects of vaccinations happen within the first 6 weeks. So 2 months is more than enough to find any of those large or, you know, common adverse side effects that you would find with traditional, you know, vaccines. So I think those are 2 points that people that I've seen come up in the news a lot, that people are afraid that, well, did we take shortcuts? We didn't. We had a lot of support and the science was robust. So they were able to do this in sort of record time, but the science is sound. And I think that's the message that people really need to hear.
So I'm gonna, oh, go ahead. I'm sorry, because
Oh, it's okay. I was just going to add, I think that was great. And that's so important to recognize that that was the whole purpose of Operation Warp Speed, is to get it done at that speed. And the only additional thing was in phase 3, they actually had a much larger study population than most other studies. So when we think about safety data and do we have enough sample, you know, large enough sample size, we actually had a bigger sample size than most other vaccine studies in the phase 3, third phase of the trial. Yeah.
And so thank you very much. I am going to go on to a different question. And one of the things that we had that came through was, is this actually really a pandemic? One of the statements was that 80% of people that are dying from COVID are over 80. So is the vaccine actually necessary? And I'm going to open that up to, I want to show something first, because again, this is data driven. This is information from the Heritage Foundation, which is a non-political group that just collects data. And
The
what you can,
Heritage Foundation is a conservative organization.
oh, well, thank you.
It is a very political organization.
So, well, even better then, even better, because as a conservative organization, you can see that 93% of the deaths from COVID, and this is as of February 17th, occur in people over 55. That's in the US. If you look at California, it drops down to 50. 90% of the deaths are in age 50. We have in the United States, in the states that actually submit data with regard to children, and not all 50 states do that. There are currently right now somewhere between about 300 to 400 deaths in children in only 20 states reporting. So children being actually under 6. So this is I mean, people are dying in a rate that is not per the person's statement, but again, 93% of deaths from COVID are in people aged 55 or older nationwide, 50 and older in California. And then I think Dr. Gupta, if you can just give us a real simple, what's the definition between an epidemic and a pandemic?
Yeah, sure. Yeah, this is a really interesting question. Actually, there's so many points that we can talk about with this. Just, I mean, definitions wise, you know, an epidemic just is a disease that affects a large number of people within a community or a region. And then it turns into a pandemic when it crosses borders. So, you know, people talk about the P in pandemic stands for passport. So if the disease has a passport and it can get to different parts of the world, and it becomes a pandemic. So certainly COVID falls into the pandemic category. But I think the bigger misconception there is that this disease doesn't kill young people. There are a couple of points. So one is that's just not true. So if you look at all-cause excess mortality in the United States for the past year, and I can actually share my screen so you guys can see the paper. I'm not going to, you know, bore you with all of these specifics here, but I don't know if you guys can see this paper on your screen. But this is just a research letter that was published in JAMA back in February. And it just looked at all-cause excess mortality in the 25 to 44 category. So even younger than that 55 number that the Heritage Foundation is talking about. And what they found is that the excess mortality from COVID in this cohort was higher or equivalent to the number of opioid deaths in this cohort. So what that's telling us is that young people, 25 to 44, do die of COVID, and they're dying at higher rates this past year than they do in historically. So it absolutely does kill young people. COVID doesn't care how old you are. On top of that, I think in the ICU especially, we talk a lot about death not being the only metric of interest. So I can tell you that one of the first patients I intubated back in March was a 22-year-old obese male with ARDS due to COVID. And he was intubated for 7 days. He had a very long recovery. was not able to go back to work for many, many months. While he survived, his life was fundamentally altered by this disease. So just because you're not likely to die being young from this disease, you can still have significant morbidity. You can still have significant, we talk about long COVID, post-ICU syndrome, prolonged respiratory rehab is often needed after you're intubated for 6 or 7 days, and that's the short end for COVID. We have people that are intubated for months because of this disease. So death is not the only metric that we should be looking at when it comes to measuring outcomes from this disease.
Thank you. Dr. Nand, can you talk to us about the risk versus benefit of getting the vaccine?
Yes. So we've all heard from the beginning that although it's not the young who are, it's not the young who are dying necessarily, the older are dying, but it is the young who are taking it home to our parents, grandparents, to our loved ones who might be immunodeficient from whatever cause, or to our young, presumably healthy friends who have undiagnosed immune deficiencies. So not only are we protecting ourselves, we are also protecting those we love the most. With the very few side effects and mainly just local symptoms that patients have with the vaccine, there's really no reason right now to not take the vaccine when it's offered to any population. There are certain patient populations where we're still waiting to see more data on, but for the most part, when we get vaccinated, we're protecting ourselves, but more importantly, our older parents. I think, as already was mentioned, COVID doesn't care how old we are, and there's absolutely no test right now, no way to predict which young person is going to do poorly from COVID and which ones aren't going to do poorly. So why take the risk when we have a vaccine, that multiple vaccines at this point, that have very few numbers of side effects and especially very few severe adverse events? There's no reason to not take the vaccine.
So I want to go off of that and answer a couple of questions that have been posed. You talked about the immunocompromised. So the question for both you and Dr. Gupta would be, are there any issues that an immunocompromised patient would need to know that might be different than a non-immunocompromised? And I will admit to the panel that I did ask this of my husband because as a rheumatologist, his patients are immunocompromised. So the first thing he said to me was, what is the compromise? Are you just taking steroids? Are you pregnant? Are you diabetic? Do you have rheumatoid arthritis or lupus? And so I will let you all answer. I'm going to make sure that I'm asking the question. So there's a couple of different points. What, if anything special, should someone immunocompromised know when making decisions about the vaccine? And the second part that I'm actually going to answer is, were immunocompromised folks included in the vaccine trials? And the answer to that second one is yes. There were rheumatoid arthritis, there were patients on chemotherapy, and there were other numbers of lupus patients and things like that. They were not all broken out, but they were not excluded on purpose. And there were no side effects in that population that was greater than any other population that was tested. So I'm going to open it up, I'm sorry, to Drs. Gupta and Nand about if there's anything other special that an immunocompromised person needs to know.
I think we see a lot of complications from natural infection in COVID patients, but as you already mentioned, they were all included in these studies. And as far as I know, there's no special precaution except for those severe allergic symptoms that patients, very few, but a few did have it, which are really related to the ingredients of the vaccine rather than necessarily their underlying immune conditions, immune deficiency conditions. I have a resource here. Let me see if I can specifically answer, but Dr. Gupta, if you want to take a stab, I'll look at my resource quickly.
Sure.
I think, yeah, Garima, I think you had a slide that showed the ingredients. Is that,
I think Dr. Hermel actually had
had the slide that showed the ingredients?
in that slide.
But which are the, excuse me, what are the things that patients are reacting to? Is it just one of the the lipids, or is there anything else that they should be concerned about?
Let me share. Oops, no, that wasn't it. Let me share my
It
screen because I
appears to
did.
be the polyethylene glycol component.
Yeah, exactly.
Exactly.
Mhm.
Yeah.
Okay.
And that's the only one where it's recommended to talk to an allergist because there might be some challenges that they can do with similar products before a person gets vaccinated.
So, oh, go ahead, Garima.
And I think with some of the biologics, there's a timing component that I've read about. I don't know the details, but if you're on some of the, the monoclonal antibody types of drugs for an immunocompromised, and you're immunocompromised because of that, it's not that you can't get the vaccine, but your physician should be able to help you figure out the timing of when to get it. So that's something that definitely affects people who may be on these certain medications, but otherwise I don't think there's any special precautions for people on steroids or pregnancy. Certainly it's pretty well understood that there haven't been any adverse outcomes in pregnant women who've been vaccinated matched to those who have not been vaccinated.
And actually, again, speaking for my husband, and I apologize because he's actually giving vaccine right now on campus, but he said as long as the vaccines are not live, which they are not, then even timing around the biologics doesn't matter. So that answers that as well. But that brought up another question. Oh, shoot. Now I have to find the question. I apologize. So if I've had a bad reaction was not defined to a vaccine, or if my family has had a bad reaction, I'm a little hesitant to take this vaccine. And so I know it's difficult to answer or comment on that if we don't know what bad reaction means. But let's assume not anaphylaxis. Is there anything that I should be worried about with this vaccine?
The short answer is no. There is actually just recently CDC put out a paper. Let me see if I can find it for you guys. And they looked at like the first month of data, of safety data. I think I have it here. So this is like the first month in the United States, and you can see that of course we vaccinated just tons of people in this first month. And if you look at the primary side effects or the adverse side effects, so let's see here, over 8 million vaccines. No, that's not it. Oh, here we go. 13 million vaccines in that first month were administered. They talk about adverse events. So 90% of those adverse events were non-serious, and then 9% were considered serious. And there were 113 deaths, but those deaths were not related to the vaccine itself. And if you look at the side effects they're reporting, I mean, these are very, headache, fatigue, dizziness. There were very few reports of anaphylaxis, actually, 62 reports of anaphylaxis that were confirmed. And there's a section down here. So this is where I think the money is for the people that are worried about anaphylaxis, which is really the main side effect of concern that came up in the early days when the vaccine came out. So and this includes Pfizer and Moderna. So 4.5 cases per million doses administered. And this is on par. This is not that different from other vaccines that we would never, ever, consider not getting. So the inactivated influenza is 1.4 per million. Pneumococcal is 2.5 per million. The live attenuated herpes zoster, 9.6 per million. Also, you can treat anaphylaxis. All these people that had anaphylaxis due to the COVID vaccine, they didn't even get admitted to the hospital. They were treated in the emergency room and discharged. Every single person we have follow-up data for did not need to be admitted. There have been no deaths from the vaccine due to COVID. So, I think certainly we worry about side effects, but what we've seen from the trials and from the initial effectiveness data as we administer the vaccine is that this is exquisitely safe.
Thank you. Is there any advantage of one vaccine over the other? And I'm going to make this a two-parter. We know that Pfizer and Moderna are 2 doses. Johnson & Johnson is a single dose, although they're developing a 2-dose as well. And so is there an advantage of one over the other? And then one question specifically to Dr. Hermel was, if I'm doing a lot of traveling, does it matter what vaccine I have? Or the person was told to wait for Johnson & Johnson specifically if they're doing a lot of overseas travel.
My take, and I'll rely on Drs. Gupta and Nand on the actual numbers of efficacy. My take was that the mRNA vaccines, the Moderna and Pfizer vaccines, were slightly more efficacious than the J&J vaccine. But obviously a single dose vaccine is going to be more optimal than a double dose. My take on the 2 mRNA vaccines is purely logistical. If you don't need a -80 freezer to store your vaccine, it's a lot easier to administer the vaccines. So that's why I would lean to Moderna only if I were a hospital administrator, but as a clinician and an immunologist, they're equivalent.
And I think the most important thing is whichever vaccine is available to us at that time, because we don't know which vaccine will come into our community and which one we will have access to. But the most important thing is being vaccinated. Something is better than nothing. I guess it's more important to have some sort of protection. And we keep hearing about these variants that are circulating in other parts of the world. But from the most recent, from just last week when I was on a CDPH call, we had a little uptick of the strain that was found in the UK and California. However, for the most part, that strain hasn't taken over as the majority of the cases yet. The other 2 strains found in Brazil and South Africa, we have not, that isn't a predominant strain here also. So at this time, we don't have to wait for a 3rd vaccine or a 3rd booster or anything like that, because the strains that are circulating are amply covered, or the strains that are currently circulating, the available vaccines will amply protect us against.
Thank you. So one of the other concerns that came up out of the chat is we don't have long-term data right now on what can happen with this vaccine. And there are patients that are concerned about the safety of the vaccine long-term. And that may make them hesitant to take the vaccine as well. And if any of you want to comment on that.
Can I show you what the CDC has to say?
Sure.
I'm not going to read the slide. You could all see that yourselves. But, from an immunological standpoint, there are no long-term side effects from vaccination. You either see it right away or nothing. I don't see anything, I'm sorry to use the term, I don't see anything magic that would suddenly make this vaccine more dangerous 10 years down the line compared to any other vaccine that's been administered. And certainly compared to, we certainly know that natural infections have long-term consequences.
Thank you. I have one other question. Thank you very much for the questions. I have one other question about how do we know when we've generated herd immunity, and if you can address what that actually means as well. Let me see if I can find the actual question.
Yeah. Herd immunity is dependent upon the virus. Measles requires up to 90% plus of the population to be immunized. But with, I think, mumps, it's less. And I don't know the numbers for the other, viruses like chickenpox, rubella, etc.
I haven't heard anything about that in terms of the COVID, SARS-CoV-2 virus and the vaccine yet. But we need a lot more people than what we have so far.
What we have. And I think to answer, and correct me if I'm not answering this in enough of a layperson's language, but if you have 100 people in a room and 80 of them, 90 of them have a vaccine, what is the likelihood that the other 10 to 20 of them will get it? Well, there's the answer is with herd immunity, the theory here is there's not enough people that are going to pass it, the infection, because they've all been immunized. So the more people that are immunized, the more the population is protected from whatever infection we're talking about, because there aren't a lot of people that then pass the infection on. And that is as non-scientific as I can put herd immunity, in a sense. And then again, correct me if I'm not saying that right.
I think that's perfect. It's about breaking the chain. It's if you've got the row of dominoes, it's about what can you do to take one domino out so they don't keep falling. And one of the things that is now emerging, data that's emerging, is about asymptomatic infections and the ability to transmit the virus if you're vaccinated. The initial studies really did not, that wasn't their goal. The goal at that point was to look at severe infections and, trying to reduce the burden on hospitals. But now that we're vaccinating, millions of people around the world, and some countries are doing better than others, there's more data coming out about asymptomatic infections as well. So I think the big one is it's out of Israel. That was I think it may be preprint right now. It's a Lancet study that looked at healthcare workers, and they looked at the rate of asymptomatic infections. And it looks like the initial data, again, we haven't proven this definitively, but it looks like most of these vaccines do, in addition to reducing severe infections and symptomatic infections, do reduce the amount of asymptomatic infections, which is what we all expected to find. We just didn't have the data to support it. So that's a really good sign in getting to that, herd immunity point, whatever that may be for this virus. We just don't know. And if you're asymptomatic, if you can't, the other thing is if you can still get it, can you transmit it? It looks like the rates of transmission in vaccinated individuals is also much lower. So these are all really, really good signs in terms of getting to herd immunity, whenever that may be, that, these vaccines, not only do they stop you from getting sick and going to the hospital, but they stop you from getting the infection, they stop you from spreading it to, people you love, people you live with, and just, when you're out and about in the community.
Thank you. Going in a little bit different question. If I have tested positive for COVID in the past, do I need to have both doses of Moderna, or is one dose enough?
So yes, the recommendations currently are to get both doses because some of the studies have shown that the immune response or the level of protection from natural infection is expected to be much lower than what the protection is after vaccination, because we actually know the amount of perhaps response that we expect with the vaccine, rather than every person's immune response or protection level is different after natural infection, but vaccine has a little bit more control. So yes, they should get both the vaccines at the data that we have so far.
So does it mean anything in particular if I have very little response and I really don't have anything except maybe the rash, but somebody else who is my gender and age gets chills and fever from getting the vaccine? Do individual reactions mean that one person has more immunity than another?
No, and that's the case with actually many infections as well. We can have people living in the same household and have recurrent skin infections from Staph aureus, and the same people in the household have never had that complication. So everybody's body responds differently to medications, to exposures, and vaccines are no different. So no.
And this was studied in the phase 1 and phase 2 trials for all these vaccines, where they looked at antibody levels in patients who received the vaccine, and while they had various or varying degrees of reactions, they were all immune at the end. So that they all had protective levels of neutralizing antibodies regardless of the amount of reaction they had.
Do any of you know whether or not we're going to need to continue to be boosted for this vaccine, similar to what we see with influenza? Yes, Dr. Hermel.
Everything depends on the variants. Okay, that's the wild card in the deck. On the positive side, it just means that the vaccine manufacturers could just easily create the new mRNA that contains the variant with the mutation, and have it's a double dose, so you're protected against the current strain that's running around, and then let's say the California and Brazilian variants. It's still an unknown question. The coronavirus has mutated at a rate much slower than the influenza viruses do. So I'm cautiously optimistic, but I think that it can be dealt with. And I think that we have the technology, this particular technology, if applied in a rigorous fashion, could eradicate the SARS-2 coronavirus from the world, just like we did with smallpox.
So it may end up being something like measles, where we all thought we only needed to get one MMR, and then all of a sudden we had an outbreak in college students in the late 80s, early 90s, I think. And then now we give 2 doses to boost the immunity. Or again, but that's because it's a slow mutator, or we may see, like the flu vaccine, that we have so many other different strains. And again, the strains right now, and again, correct me, do not relate to severity of disease, but ease of contraction. So some of the strains are more infectious than others, but the patient's symptomatology is identical if they get
Well,
this strain or this strain, or so
yeah.
Go ahead.
Go ahead, Dr. Nand.
Oh, sorry. So there's some preliminary data coming out of the UK actually that that's probably what you were going to say, Dr. Gupta, too. Yeah, is they are seeing more severe, these patients, because they are seeing more severe cases of these UK variants. So whether it's because it was more infectious, so more people got infected, but what we initially thought that the severity was not any different might not be the case when we finally see that data from the UK.
Okay, so in that case, we would be looking at maybe at a UK booster or something like that?
However, I think the currently available vaccines actually have protection against the UK variants.
Yeah.
So
And
far, all the
also, I
data
mean,
is-
variants emerge, mutations happen if the virus is replicating and widespread in the community. So the vaccines are still the answer. You know, the more people that are vaccinated, the fewer infections we have in the community, the less chance that virus has to replicate. That's how we're going to knock out mutations and new and clinically significant variants.
We had an early question on that was a little bit more technical, which I'm going to ask right now. So I've been given the shot. What protects that mRNA from being destructed? I can tell you, I can guess the answer, and in the first place, and then how does it get into the cell and not be destructed into the cell?
The liposome surrounding the mRNA prevents it from being destroyed by RNases that might be found in the blood. Once it's in the cell, it's just like any other messenger RNA. Like if this particular messenger RNA was destroyed, then we wouldn't be able to have our cells make proteins.
So it's carried in the bubble,
Yeah.
like you had said before. That's what protects it from being destroyed in the first place.
Yeah.
Once it's in the cell, then this, what I had referred to, this zippering, you know, it's read, the message is transferred, it's somewhere else, it's read again, it's transferred somewhere else, will occur without it being destroyed.
That is correct.
I don't know if that answered the question, but
How does it get on the cell surface, Evan, after that? How does it get exposed to the immune system
Meaning
inside the cell?
Okay, meaning the spike protein?
Yeah, assuming that it has to get exposed to our immune system and it's inside the cell, how does it get out to be exposed and then become epigenetic?
Oh, the protein will be broken down by the normal protein processing machinery of the cell. Then, you know, proteins have a finite life in the cell. They go to recycling, and then they are turned into peptides. Those peptides are loaded onto our MHC molecules and then displayed to our T lymphocytes, which are on the prowl for peptides from non-self proteins. This is the whole reason why our adaptive immune system works, is to be able to differentiate self from non-self. So, you know, Greg from non-Greg. And once those T lymphocytes recognize that there are these peptides from the spike protein that are foreign, they will then give the orders to B lymphocytes to make antibodies to the protein. We have these, we make billions of B lymphocytes daily that can make antibodies to basically anything. Right now, in all of your bodies, you have B cells that can make antibodies to Ebola virus, to Zika virus, to compounds that have never existed before in the history of the universe. But if Miriam Gawkin goes into her lab and makes such a compound and then injects it into, say, Roman, right? Within a week, he'll make antibodies to that. So it's this combination of, because we have so many of these B cells that can possibly make a potential antibody, that we do have a pool of anti-spike protein antibodies ready to go as soon as they get the order from the T lymphocytes that see that peptide from the spike protein. It's not necessarily that the spike protein goes to the surface, but that may be, because I don't know the sequence of the spike protein to see if it is expressed on the cell surface to then produce the neutralizing antibodies that we want to make.
so I think a lot of the confusion with this vaccine is because we're saying RNA and DNA. And so we tend to think of it as cellular structure and mutation. And again, I hope that it's hard to put it in lay terms, but I hope that we explained enough that the message of create antibodies against the spike protein is what's being sent by the vaccine. And that it's not getting into the nucleus of our human cells that tell our human cells alter. Okay, that's cancer. Cancer does that. The vaccine does not. What I've got, and I know that we're a little bit over time, but if anyone else has any questions, please let us know right now. Just type it into chat. I want to thank Drs. Hermel and Gupta and Nand for volunteering their time to come in and just talk. And for those of you that submitted questions earlier, for those of you that submitted questions during the chat, this has been recorded. We are going to try to make this available to those of you that don't have Canvas. So I'm going to ask the provost to post this somehow so that those of you that are in HR, those of you that are not faculty or students can access this later. And in addition, Dr. Gupta had provided, again, the speakers had been given the questions ahead of time, most of them ahead of time. And so I have a list of references from Dr. Gupta for anyone that wants to look those up or have them for reference. I have Dr. Hermel's slides as well. And we'll make those available for everyone as well. Are there any last minute questions before you all go?
If there are none, I'd like to address the concern about infertility.
Yes, talk to me about infertility.
So a former scientist who worked at Pfizer, I believe, raised the issue that the vaccine might make people infertile. His concerns were based on the idea that there are sequences in the spike protein that have some homology to a protein called syncytin 1, which is found on placental cells, but the sequences are too small to be immunogenic. And during the clinical trials, a number of women got pregnant. So his concerns, and the person in particular who raised these concerns was a vaccine denier or a vaccine doubter. He was an epidemiologist, not an immunologist. And so his concerns were proven to be false.
Thank you. All right, again, I want to thank the three of you for participating. I hope this forum was okay for the three of you. And a special thank you again to Dr. Nand, who is joining us from Stockton, from the Touro University Medical Group. And then again to Drs. Gupta and Hermel, who are on faculty here in Vallejo. If you do have any other questions and you didn't want to bring them up, you're welcome to email me and I will pass them on to the group if you have other questions or concerns. But the main thing we wanted to do is alleviate any concerns you may have about receiving the vaccine. We are all, for us from a science background, are really going to tell you, go get this vaccine. It is the best way you can protect not only yourself, but your family members from contracting the disease. And whatever vaccine you can get is the one that is the best one for you. Take it when you can get it, is the way we're saying. I'm running an independent study. This is off topic. I'm running an independent study of my own family who have now all received all of the different vaccines. So my husband got Pfizer, I have Moderna, my sister got Johnson & Johnson. We're running one clinical study. So we'll get back to you in a year and tell you how it worked.
If you could get me an ELISA kit, I'll be happy to assess your IgG titers.
I don't have any because I didn't react. No, I'm teasing. That'll work.
So
That'll
Chris is
work.
opposite as well, Gail. He's Moderna, I'm Pfizer. And you know what? If you want to do something, Evan, we volunteer our blood.
Yeah, we can do it.
Yeah, we do it. If you,
I can't get my sister. She's in Colorado. You're on your own for that one.
Yeah,
We have to get J&J out here.
we would, that might be interesting if we have enough people who've done more than one. You know, and
That's
within-
funny. Yeah. But all right. Thank you all. And again, as soon as this gets posted, however it gets posted, we will send out another mass email to everybody. So thank you, and have a really, really good afternoon and weekend.
Thank you.
So
Thank
thank
you.
Well, stay safe.